Now we're going to go back into the process of spermatogenesis. Again, we've already looked at it some, and we looked at the overall cross-section of the seminiferous tubule and the process of spermatogenesis there. We're going to look at it in a little more detail here. First of all, spermatogonia, remember, were those cells out at the perimeter of the seminiferous tubule. They divide by mitosis to increase the number of cells that can undergo the process of spermatogenesis. So there are a number of spermatogonia, and their numbers keep increasing at a fairly regular rate. But some of those cells, and the spermatogonial cells, when they divide, instead of just adding to the supply of spermatogonia, they produce cells that move toward the middle of the seminiferous tubules, and those are your primary spermatocytes. And they are also diploid cells. So they haven't started the process of forming haploid cells yet. So this is the cell that initiates the process of meiosis here. So these are diploid cells. Each of those then would undergo this process of meiosis. So the first division from the primary spermatocyte leads to the secondary spermatocyte. And these cells are haploid, so going from the total number of 46 chromosomes down to 23 chromosomes. The chromosomes are all paired here in homologous pairs, and in this division right here between this stage and this stage, those homologous pairs are divided so that each cell just gets one representative from each homologous pair of chromosomes. So that's the first meiotic division, sometimes called the reduction division, because we've reduced the number of chromosomes. The next division, the secondary spermatocyte, each of those will divide to form two spermatids. In this layer here, these are still haploid cells, but the two halves of each chromosome have pulled apart in this case. So they both have the same kind of genetic information in each of these cells. These would have a different complement, but each of these two would be the same over here on this side. So we get these four spermatids to result from a single primary spermatocyte, and these undergo a transformation, a change in shape. In the process, they lose a lot of this cytoplasm, this kind of what we describe as being sloughed off, being discarded, and we'll come back to that idea a little bit later. But anyway, it undergoes a change where this cell has kind of a little blob of cytoplasm off the side as it becomes a sperm. And that little blob of cytoplasm that comes off there is called a residual body. So each spermatid loses its residual body in the process of transforming into a mature sperm, or we can call those spermatozoa. Either way. So those are the stages of spermatogenesis. Cells divide mitotically. Some of those cells become primary spermatocytes that enter the next meiotic stage, the first meiotic stage of division, to go from a diploid to haploid state. Then those haploid cells divide again to form spermatids. Spermatids transform into spermatozoa. Now, earlier we talked about the layering of materials in the seminiferous tubule wall. We had peritubular cells indicated by brown in this illustration. Then we had the spermatogonia out against the wall. They divide and produce primary spermatocytes. Those will eventually divide and produce secondary, and those divide and produce spermatids. But we want to talk about Sertoli cell functions here. So in the green here are the Sertoli cells. the supporting cells inside of the seminiferous tubule. Some of the roles or functions of the Sertoli cells that are in here between these other cells that are in the process of spermatogenesis are one, the concentration of testosterone. So testosterone is produced by the interstitial cells of Leydig, what we call Leydig cells in the interstitial regions of the testis. They produce testosterone, but a lot of that testosterone is concentrated in these cells here. And that concentration of testosterone around these developing spermatogonia and primary spermatocytes is part of an essential element of spermatogenesis to cause them to undergo that process of sperm formation. So that's helpful to have that concentration of testosterone surrounding these cells. Next is what we call the blood-testis barrier. The blood-testis barrier. These cells that divide here, these are like normal body cells, but after they divide, they don't have the same complement of, well, at least in the haploid state, once they get to the haploid stage, they don't have the same genetic information exactly as the parent cells do. They only have half of the information, so they may seem a little different to the body's immune system. So we don't want the body to come in contact with cells that are different than normal body cells. So we have this barrier across here we call the blood-testis barrier, where each of these Sertoli cells makes tight contact with neighboring cells and essentially seals off these spermatogonia over here from all of the subsequent stages of sperm formation in the seminiferous tubule. So that's what they call the blood-testis barrier, this layer across here where all these Sertoli cells kind of seal off the luminal cavity and the more advanced stages of spermatogenesis from the outer portion here. So that's number 2, blood-testis barrier. Number 3 is nutrient delivery to these cells. These cells undergo some growth still, even after they divide here, so sometimes your primary spermatocytes are fairly large. So anyway, that growth requires nutrient delivery. They nourish those cells. Number 4, another function of the Sertoli cell is recycling residual bodies. We talked about residual bodies earlier, how there's cytoplasm sloughed off of spermatids as they become spermatozoa. So all of that cytoplasm, the nutrients and materials in there can be recycled and become part of that nutrient delivery. Number 5, inhibin secretion. Inhibin is a hormone, and it sounds a little like inhibit, and its function is an inhibitory function so that it feeds back to the brain locations and pituitary gland to inhibit the secretion of follicle-stimulating hormone and luteinizing hormone from the pituitary that causes more testosterone release. So once you have plenty of testosterone, the Sertoli cells have a way of communicating that back to the body to let it know that there's no longer a need for more follicle-stimulating hormone or luteinizing hormone. We can slow down the production of testosterone. So that's what inhibin does. One thing that you should know, and that I often put on an exam as part of the test over male reproductive system, is the path of sperm. So from its origin, all of the tubules and passageways that sperm will pass through on its way through the system. So here's a list. We've already been over about all of this, I think, starts in the seminiferous tubule. The final part of each seminiferous tubule is that straight tubule, sometimes called the tubuli recti, those straight tubules in there. Then it enters into what we call the rete testis. Rete, remember, the network of passages that lead into the efferent ductules that bridge between the testis and the epididymis. Those several passageways that lead directly from the testis to the epididymis are the efferent ductules. Then you go into the epididymis, which is a large coiled tubule on the outside of the testis there, and it leads eventually into the ductus deferens at the very bottom of the testis in the scrotal sac. That's where it begins. It then passes up into the body cavity and circles around to a position behind and below the urinary bladder, where it connects to the urethra. That last segment of the ductus deferens is called the ejaculatory duct. It is between the seminal vesicle, between the seminal vesicle and the urethra in the prostate. So it actually passes through the prostate, that ejaculatory duct there. Next is the prostatic portion of the urethra. So the ejaculatory duct leads into the prostatic urethra. Prostatic urethra then goes into membranous urethra, and then finally penile urethra. So those are the 10 different passageways that sperm will pass through on the way out of the body. Next, we're going to look at the hormonal regulation of sperm development. This is going to be important again when we look at female reproductive cycling. So many of the same hormones are involved in regulation of sperm production as well as the female reproductive cycle. Starting up here at the top of my diagram, this is in the floor of the brain. This is the region called hypothalamus of the brain. There are cells in your hypothalamus that produce a hormone that will stimulate your pituitary, will circulate through the blood between the hypothalamus and the pituitary. This hormone is called gonadotropic release hormone. Commonly known as GnRH, gonadotropic release hormone. It's produced by the hypothalamus. It's sent to the pituitary. So this is your pituitary. Here. The pituitary, in response to that hormone, will release two different kinds of hormones. One of them being what we call luteinizing hormone. Or LH is the common abbreviation for that. The other that's released is follicle stimulating hormone. Or FSH, follicle stimulating hormone. Both of these have their effect in the testes, but in different tissues of the testes. The affected tissue for luteinizing hormone, the target tissue there, is the Leydig cell. Of the testis. Follicle stimulating hormone, along with testosterone produced by the Leydig cell, will affect sperm development. But the Leydig cell is relayed through the Sertoli cell, to affect spermatogenesis. So this loop along here, this is all testosterone. So testosterone and follicle stimulating hormone both have their effect on spermatogonia and primary spermatocytes to stimulate the process of spermatogenesis. Now, earlier we mentioned that there's a hormone called inhibin released from the Sertoli cell. The Sertoli cell's inhibin inhibits the production of follicle stimulating hormone. So the more active the Sertoli cell is from the release of testosterone, the more inhibin it will produce, and that will reduce the amount of follicle stimulating hormone. Testosterone also has an inhibiting effect, and testosterone then reduces the amount of luteinizing hormone released from the pituitary. So both of these hormones, testosterone and inhibin, have an inhibitory effect and reduce the amount of both follicle stimulating hormone and luteinizing hormone sent down the pipe through the blood to the testis.